Buenas Danhafide. My name is Laura Cacer and I'm from Guam. And today I'm going to talk to you about my research over the summer on the mollusks of Kawainui Marsh and the parasites that they carry. That's it's just it. So, Kawainui Marsh was once part of an ancient caldera as part of one of the three volcanoes, the youngest of the three volcanoes that form the island of Oahu. Um, Kawainui Marsh itself is about 830 acres big, but the area that our study took place was right here on the map near Ulupo. Kawainui Marsh is a very important resource, both environmentally and culturally. It is a place where many of the ancient Hawaiian mo'olelos or legends took place. Oh, my bad. Kawainui Marsh is also a protected area and home of many many endemic bird species. Kawainui Marsh was traditionally used by ancient Hawaiians for food production. They grew a lot of kalo, they produced fish, as well as breadfruit and bananas. In modern times, the area is being refurbished by Ka'uluakalana to produce many of these same crops. But the difference between ancient times and nowadays is that the ecosystem has changed drastically, and this is due mostly to invasive species. Invasive species, as defined, or as we're going to use it in this research project, are species that have been introduced to a place and are not native here, and things that cause harm to the place. A couple examples that I'm sure you guys are familiar with are the rats. They're a huge vector for disease and are a huge pest, both in households and agriculturally. Euglandina, the rosy wolf snail, as it's commonly known, has been a huge driving force in the extinction of our native snails here in Hawaii. And Pomacia caniculata, which totally altered the ecosystems that they enter. Speaking of snails, one of the reasons, snails alter ecosystems in multiple ways. Of course, they come in and they compete with the native snails of an area, but in addition to their direct effects on an area, snails bring in the parasites with them when they come. They bring things like rat lungworm, different kinds of nematodes. They're a huge vector for disease and disease that affects other animals as well as things that affect people, like we mentioned, the rat lungworm. Here are a couple examples of zoonotic life cycles of parasites that affect both animals and people. And as you notice, a lot of these zoonotic life cycles need they require multiple different species in order to complete their life cycles. Trematodes, for example, oftentimes need fish and snails. Whereas Angiostrongylus cantonensis, the rat lungworm, requires snails and rats. Knowing this and the fact that things are being grown here for human consumption, it's important for us to know what kinds of things are growing here at Kawainui. So my research for the summer was to identify all of the species of mollusks here at Kawainui, as well as the kinds of parasites that they carry. In order to do that, we performed time surveys. We came to an area and people from the Bishop Museum, or the labs of the Bishop Museum, came to help us. We set a timer for about 20 minutes. We collected as many mollusks as we could find in an area, and then we moved on to the next area. Some of these mollusks come in all shapes and sizes, and I don't think I would have found a lot of them without the help of these professionals. So here are a couple examples of the ones that we found and our very delicate collection methods. So once we collected all of the mollusks, we brought them back to the Bishop Museum. And in order to identify the species of mollusks, we used morphological comparisons. We looked at their coloration, sizes, patterns, and compared them to the literature. We also performed genetic sequencing and analysis. In order to identify the parasites, it was a little bit more difficult because a lot of them are microscopic or don't have a lot of visual differences. So the first way we collected them was through snail dissections. We dissected snails we knew were infected under a microscope, and we pulled out any parasites we could find with a collection pipette. We also performed pepsin digestions, and we genetically sequenced the parasites that we did find. Our results, we found mollusks from 6 different families and 7 different species. These two are under microscopes. We found Planorbella durii, a species of ensilid, Pomacia canaliculatta, the golden apple snail, Parmarian martensi, also known as the semi-slug, Lysacatina fulca, the giant African snail, and we found 2 species of Melanoides tuberculata, one of which was a cryptic species, so we're still trying to get that sorted out. And for the parasites, we found Angiostrongylus cantonensis, the rat lungworm. You can see a couple of them here under the microscope. We found species of leeches and trematodes. It's a little bit difficult to see them in this picture, but they look kind of like maggots. We found 2 different species of trematode. Phylothalmus gralli, the oriental eye fluke, and didn't show, sorry, the liver fluke, which affects people. And an important note, finding the oriental avian eye fluke was interesting because, as we know, this is a protected area for endemic birds, endangered endemic birds. So this could pose a real issue for the conservation. Here is one of the life stages of a trematode. This is a snail dissection under a microscope. And if you guys are familiar with swimmer's itch, that's what this is. It's trematodes trying to burrow into your leg. Here's a picture of my leg after tending the lo'i this summer. Yeah. So here's a little chart of the different species that we found, the mollusk species, and the parasites that we found within the mollusks. Unfortunately, due to technical difficulties, our PCRs could not be completed and we couldn't finish a parasite screening. But as you can see, we did find a lot of different species of parasites. And some of these are very concerning. Angiostrongylus cantonensis, of course, the giant African snail and Parmarian martensi were absolutely loaded with them. We only had 11 samples of the Parmarian martensi, and every single one of them was loaded with these rat lungworms. So these are a list of the known parasites that haven't necessarily been found here, but could possibly or have been found previously in these species of snails elsewhere. This is just to say that because these snails are here, it's a possible vector for any snails carrying these to come and introduce them into the environment. This is important because, as we mentioned, things are being grown here. Food is being grown here for consumption. And that's why this research is so important. It's important for us to know what's here so that we can support Ka'uluakalana and support the sustainable ways of growing food that are being performed here. Future directions. So unfortunately, our survey methods were not the most in-depth. We could definitely spend more time here to try to compile a more complete list of mollusk species. We did find some limnaeid shells, dead limnaeids, but we weren't able to find any live species or live samples. So a more complete in-depth list could definitely be compiled. Parasite metabarcoding. The ways that we did collect the parasites could be considered a little bit crude. In order to have a more in-depth and complete list of the parasites that are here and possibly a threat to the people who are eating the food, we could do parasite metabarcoding and complete the list. And in this research, we compiled a list of the species of mollusks and parasites. We did not look into how the invasive parasites may affect native Hawaiian snails. and parasites on our table. We found things like rat lungworm, as we mentioned, and the Parmarian martensi that we found like to live in the little crevices of the kalo plant, which we eat. That may not be as big of a deal because things like kalo have to be cooked for hours and hours. But like we mentioned earlier, trematodes can live inside fish, which may not be cooked for as long. So some research should probably be done into the kinds of parasites that are here and ways to make the food safe for people to eat. Mahalo and sudu'us ma'asi for this opportunity. I've learned so much and I'm grateful to be here today. Thank you.